Hello everyone and welcome back to Dr. Wasif Academy. In previous lecture, we have discussed about what are enzymes and how they work, especially focusing on lock and key model and enzyme substrate complex. Now, think of enzyme as the precision tool of your body. They speed up everything from digestion to DNA replication. But like any high-end machinery, they only work under specific conditions. Today, we are looking at the breakdown why temperature and pH are the make or break factors for enzyme activity and what actually happens at a molecular level when an enzyme denatures. So my dear viewers, grab your notebooks and let's get started. So my dear students, enzymes are hard workers, but they are also very sensitive to their surroundings. One of the biggest factors that dictates how fast they work is temperature. First of all, we shall discuss about low temperature. At low temperatures, imagine everything is in slow motion. Molecules have very low kinetic energy, and because they are moving so slowly, the enzyme and the substrate rarely bump into each other. Fewer collisions per second mean the reaction rate is very low. But here is the key detail. At low temperatures, the enzyme is not broken. Remember it, at low temperature, enzyme is not broken. It is just inactive. It is like a car that has plenty of fuel, but the engine has not been turned on. Secondly, now let's talk about increasing temperature. Means a temperature is increasing from low towards higher. So as the temperature rises, those molecules gain more kinetic energy and start zipping around much faster. But what happens next if we increase the temperature? You get more frequent collisions between the enzyme's active site and the substrate. If there are more collisions, it means more enzyme substrate complexes are forming every second, which causes or which results the reaction rate to climb steadily. So when you are writing about temperature in your exam, always link temperature to kinetic energy and the frequency of collisions. Do not just say that it goes faster. Explain why. By monitoring that the molecules are moving more and moving more rapidly and hitting each other more often. This shows a deeper understanding of the physics behind the biology. But you should understand that what will happen if heat makes them more faster. Can we just keep increasing the temperature forever? Not quite. Let's see what happens when it gets too hot in the next slide. As we have seen that heat speeds things up, but there is a limit. Like a high performance engine, an enzyme has a sweet spot where it works best and a danger zone where it breaks down. Before that, we should look into the optimum temperature. This is the point where the enzyme works at its absolute maximum rate. That is called the optimum temperature. For most enzymes in the human body, this is right around 37 degrees Celsius. This is why your body works so hard to maintain its internal temperature. It's keeping your enzymes in their peak performance zone. But what happens if the temperature keeps climbing, keeps increasing? Things take a turn for the worse. At high temperatures, the thermal energy becomes so intense that it starts to vibrate. The vibrating the enzymes atoms very violently. This breaks the weak bonds, okay? Means this breaks the weak hydrogen bonds which hold the protein's complex 3D shape together. When this happens, the enzyme loses its shape. This is called denaturation, or you can say denaturing the enzymes. Crucially, the active site changes shape. Now the substrate, means that is the key, it no longer fits into the lock. The reaction stops, and because the protein has been physically unfolded, this damage is usually irreversible. So here is a major pitfall to avoid in your exams. That is the difference between denaturation and inactivity. Never say an enzyme is killed or it dies, because they are molecules, they are not living organisms. So we cannot use the term kill or die for enzymes. because but we use the correct term, that is denatured. Secondly, never say enzymes are denatured by the cold, because in the cold they are just inactive because they lack kinetic energy. So if you uh, warm the temperature, okay, increase the temperature, the enzymes will start working again. But if you denature an enzyme with heat, with increasing temperature, with high temperature, it is permanently broken. So always keep that distinction very clear. As temperature is not the only thing which can change an enzyme's shape, in our next slide, we will look at another silent controller, that is pH. So, if temperature is the weather an enzyme lives in, the pH is the chemistry of its home. Just like heat, the acidity or alkalinity of the environment can completely change how an enzyme behaves. First of all, we shall discuss pH stability. Enzymes are highly sensitive to hydrogen ions. Because enzymes are made of proteins, the pH levels can actually change the electrical charges on the amino acids which form the active site. So, if those charges change with the change in the pH, the delicate structure of the enzyme can start to pull apart or twist out of shape. Just like temperature, every enzyme has a unique optimum pH. This is the specific point where the active site is at its most stable and perfect complementary shape. At this pH, the enzyme substrate complex form at the highest possible frequency, and the reaction hits its maximum speed. So if we talk about examples, that is about pepsin, okay, that is in your stomach. It is important to remember that optimum does not always mean neutral. For example, take pepsin. This is an enzyme found in your stomach which breaks down proteins. Because your temperature is full of hydrochloric acid, pepsin has adapted to thrive there at this high pH, at this okay acidic pH. I should not say their high pH word. I will say acidic pH, that is around 2.0. Okay, so if you moved pepsin to a neutral environment, it would actually stop working. So in your exams, when you are discussing about pH, do not just say the enzyme breaks. Use a scientific explanation. Mention that pH affects the charges on the amino acids in the active site. Also, you should always remember that different enzymes have different optimum pH depending on where they work in the body. For example, pepsin, as we have discussed, that is a classic example to use for acidic condition. As we have seen the stomach, but what about the rest of the digestive system? Let's look at how pH requirements change as we move through the body. While the stomach is acidic, other parts of your body are much milder. For example, take amylase. This is the enzyme in your saliva. Amylase works best in roughly neutral conditions. Its optimum pH is around pH 7. Now, take a closer look. The optimum pH for pepsin is 2.0. It means that works best when the pH is 2. But amylase works best at pH 7. This is why the chemistry of your mouth is kept very different from the chemistry of your stomach. It is all about providing the right home for the right enzyme. But what happens in the danger zone? If the pH becomes much lower, meaning too acidic, or much higher, meaning too alkaline, then the trouble starts. These extreme conditions disrupt the hydrogen and ionic bonds which are holding the protein's 3D structure together. Just like with high heat, means with very increased temperature, the enzyme's shape is altered, it changes. So once the active site changes, the substrate can no longer bind. The enzyme has been denatured and the reaction rate will drop drastically or stop completely. It is a physical change that the enzyme usually cannot recover from. In your exams, you will often be asked to analyze a graph. We will also discuss a graph after completing this lecture. Okay. But what I want to explain here, that you should always identify the peak. Find the very highest point of the curve and drop a line down to the x-axis to find the exact optimum pH. Do not just say the line goes down. Use a scientific reason. Say, at extreme pH values, the enzyme is denatured, causing a steep decline in activity because the active site shape is lost. And remember, every enzyme is a specialist. Never assume all enzymes like pH 7. Okay, always look at where they work in the body. We have discussed this scenario regarding pepsin and amylase. So as we have used the word denatured several times now, but what is actually happening at a molecular level? Why does heat causes a protein to lose its shape? Let's zoom in on the atoms themselves. First of all, we know that enzymes are proteins. The foundation of everything is this. Enzymes are proteins. They are not solid blocks. Okay, they are long, complex chains of amino acids which have been folded into an intricate three-dimensional shape. Think of it like a very specifically folded piece of origami. Okay, and in biology, shape equals function, or you can say shape dictates function. That precise 3D geometry, especially at the active site, is held together by many weak chemical bonds. We are talking about hydrogen bonds here and ionic bonds also. Individually, they are weak, but together they hold the protein in its perfect working shape. So when the temperature rises above the optimum level, the atoms inside the protein begin to gain a massive amount of kinetic energy. They start to vibrate violently. Imagine those atoms shaking so hard that the delicate hydrogen bonds simply cannot hold on anymore. They snap. So once those bonds break, the protein unfolds and loses its unique shape. It becomes denatured. It's like that piece of origami being flattened out. You cannot just fold it back perfectly once the structure is ruined. So as we have seen how heat shakes a protein apart, but pH works a little differently. It's more like a chemical tug of war, which pulls the enzyme out of shape. When the environment becomes too acidic or too alkaline, there is a sudden change in the balance of hydrogen and hydroxide ions. Okay, these ions carry electrical charges, and because your enzyme is a protein made of amino acids, these ions start interacting with the charges on the protein itself. This interference disrupts those essential ionic and hydrogen bonds, and without those bonds to hold it together, the protein begins to unfold, losing its shape, specific three-dimensional shape. The question is, what is the final result of all this unfolding? The answer is, the active site is damaged. It is no longer complementary to the substrate. The key no longer fits the lock. At this point, the catalyst simply stops working. The reaction halts, and in most of the cases, this change is irreversible. So once the docking station is gone, the enzyme's job is over. So if you are aiming for those higher marks, you must master what I call the trio of denaturation. In an exam, never just say it stops working. You must follow this three-step logic. The first one is bonds. State that the high temperature or extreme pH breaks the internal bonds like hydrogen bonds. The second point is shape. State that this physical change alters the shape of the active site. And the third point is fit. Conclude your answer that the substrate no longer fits because it is no longer complementary. So if you miss any of these three links, you miss the full marks. There's a chain of events. Make sure you tell the whole story perfectly. As we have covered a lot of ground today about enzymes, So to wrap up the theoretical part of our lesson, let's bring everything together into one clear picture of why enzyme specificity is the backbone of biology. Specifically, specificity is not just a detail. It is the foundational principle of biological catalysis. Okay, it is the rule book that dictates how an enzyme chooses exactly which molecule to interact with and which ones to ignore. Remember, the enzyme is a masterpiece of protein folding. Because of its unique sequence of amino acids, it forms a specific 3D shape containing that vital pocket, that is the active site. Okay, the geometry of this site is fixed and very precise. Number three is the interaction. This is where the key comes in. Only a specific substrate has the complementary shape required to dock into that active site. So when that perfect geometric fit happens, we get the enzyme substrate complex and the reaction proceeds. Number four is here is before discussing point number four, I have a question. Why does the cell do this? Means why this specificity all this happens? Why not have an enzyme that does everything, means a super enzyme? So the answer is control. Okay, the answer is control over metabolic processes. By being specific, enzymes ensure that your metabolism is very well organized. It prevents a chemical chaos where every reaction happens at once. Instead, life is a perfectly timed and regulated series of events. So, as a tip for you, when you are asked to summarize specificity in an exam, do not just stop at the log and key definition. To secure high marks, you must link to the why. Okay, you must link to the why. So you should master this chain. Okay, state that specificity is due to the unique active site being complementary to specific substrates. Link this to the result that this allows the cell to manage complex metabolic pathways in a controlled and efficient way. Mentioning regulated metabolism shows you understand the big picture, which is exactly what examiners are looking for in top tier answers. That is the end of our lecture slides. Now it is time to put your knowledge to the test. Okay, let's move on to some practice questions to see how well you have mastered the word of enzymes. First of all, I shall give you an example of question and I shall also give you the answers. Okay, then I shall give you a few questions which you will give me the answer in the comments section. First of all, I took this question from your book. Okay, in your book you can find this question that the diagram below shows an enzyme catalyzed reaction. Okay, this is the diagram and here are three questions that state the names of A, B and C, and use the second question is use the diagram to describe what happens when an enzyme catalyzes a reaction, and the third question is explain why the model is known as lock and key model, means this model, why this model is known as lock and key model. We shall discuss these questions one by one. First of all, the question is state the names of A, B and C. A, you can see the A is substrate, specifically two substrate molecules in a synthesis reaction. Okay, two substrate molecules. And B, this is the active site. Okay, this is the active site, and in this ignore this one. Okay, and even this has not been asked. Okay, what is here? This is key is fitting into the lock, and this C, what is this? This is the product. Okay, so A is substrate, B is active site and C is the product. Next question is use the diagram to describe what happens when an enzyme catalyzes a reaction. So looking at this diagram, the number one uh, first answer, first part of this answer is the substrate molecules approach the enzyme and bind to the active site. You can see it here. Okay, second point is an enzyme substrate complex is formed because the shapes are complementary. Here you can see it. And the third point is the reaction occurs converting the substrate into a product, which is then released. So the enzyme remains unchanged. This enzyme, it remains unchanged. And the third question is, explain why the model is known as lock and key. Simply, we have discussed it throughout in this lecture, that just as a specific key fits only one lock, a specific substrate fits only into the matching shape of a specific active site. So the two shapes, these are complementary. Okay, remember this word complementary. Okay, do not ignore it. It is very necessary for you to understand. The second part is explain the following statements. The first statement is amylase digest starch, but proteases do not. The second question is human enzymes catalyze reactions at 37 degrees Celsius, but not at 73 degrees Celsius. The third question is the table shows the relative activity of a human enzyme in solution of different pH enzyme. We shall take this question later in this slide, okay? But I think we shall discuss it in this slide also. Let's see it. The first one is amylase digest starch but proteins do not. Because enzymes are specific, the active site of amylase is complementary to starch but does not fit the shape of protein molecules. If you recall this video lecture, you can understand this again, that what is difference between the activity of amylase and the activity of protease. The second question is human enzymes catalyze reactions at 37 degrees Celsius but not at 73 degrees Celsius. The answer is at 37 degrees Celsius, which is optimum for these reactions. Okay, molecules have high kinetic energy for collisions, means optimum kinetic energy. But at 73 degrees Celsius, the enzyme is denatured because the high heat, means high temperature, breaks the bonds holding its 3D shape. So it destroys the active site. This is a perfect example of it. Okay. And here is the table. Okay. This is about pH and relative activity. And there are three questions also. The first question is explain why all the reaction mixtures were kept at 35 degrees Celsius. Okay. In the previous slide, you can recall that about this table, that it was mentioned that this reaction is happening at 35 degrees Celsius. Okay, because that is a constant temperature to properly uh, respond to pH variation, that how it affects. So if we answer the first question, temperature is a controlled variable. Okay, it must be kept constant to ensure that any changes in activity is due only to the pH, making the results valid. The second question is draw a graph of the results in the table. So when drawing this graph, place pH on the x-axis, like this one. This is x-axis. Place pH on x-axis and relative activity on the y-axis. Then a bell-shaped curve that is formed. Okay, you can see the temperature temperature is rising. Okay, as the temperature is increasing, and when it reaches at optimum level, okay, sorry, not temperature is rising, sorry, pH is rising. Okay, pH is increasing. When it reaches and at optimum level, okay, then this is the maximum activity. But when the pH starts increasing, okay, it continues increasing, okay, then the reaction halts down. It moves down and then it halts. So if we describe and explain the results which are shown in this graph, the activity increases at pH as pH rises from 3 to 7. Okay, pH rises from 3 to 7. So after pH 7, the activity decreases rapidly. So how we explain this answer? The explanation is at pH 7, the active site is the perfect shape of the substrate. At extreme pH values, means 3 or pH 11, these are both are extremes, okay, on both ends. The enzyme is denatured. The charges or bonds are disrupted, so the it changes the active site shape, so the substrate can no longer bind. Here you can see that I use two words that describe and explain, because in the question it was asked that describe and explain. So what is difference between describe and explain? I shall also give you its detailed answer later in this lecture. Now that brings us to the end of today's discussion. We have explored how various factors influence enzyme activity through real-world examples and seen the vital role they play in living organisms. So, if you have any questions, please drop them in the comment section below. To stay updated and support the growth of this channel, consider subscribing. It helps these lectures reach more students like you. I wish you all the best of luck with your studies and thank you for joining me today. I will see you tomorrow for a Q&A session designed to mirror your final exam. Until then, goodbye.